Hello, everyone. Welcome to today's live broadcast titled Create Your Own Cellular Compound Target Engagement Assay, presented by Dana Haley Vicente, PhD, Senior Product Manager, DiscoverX Corporation. We are excited to bring you this educational web seminar presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. This webinar is sponsored by DiscoverX. DiscoverX is an innovative company that develops, manufactures, and commercializes reagents, complete cell-based assay kits, profiling and screening services, as well as other turnkey-based solutions for the drug discovery and life science markets. Their biochemical and cell-based assay platforms enable researchers to improve the productivity and effectiveness of their screening, lead optimization, and SAR campaigns, thus accelerating the discovery and development of new drugs. I'm Julie Simmeroff of LabRoots, and I'll be your moderator for today's event. Before we start the presentation, we have a few important announcements to make. We welcome your questions during the webinar. Since attendees will be muted during the presentation, please submit any questions by typing your questions into the questions box, which may be located near the bottom of your control panel. Following the presentation, we will address any questions that were submitted in the order in which they were received. If we run over the hour allotted and do not address your question, we will personally answer your question by email following the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know you're having a problem. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. Now I'd like to introduce Dr. Dana Haley Vicente. Dr. Haley Vicente is currently a Senior Product Manager at DiscoverX, managing cell-based assays, kits, and consumable products for target discovery, screening, lead optimization, and SAR campaigns. She has been working in the drug discovery industry for over 16 years and received her PhD in pharmacology from the University of California, Los Angeles. Please join me in welcoming Dr. Haley Vicente. I will now turn the presentation over to her.
Thank you, Julie, and thank you everyone for joining. Today's seminar is creating your own cellular target engagement assay. So this is more like a DIY or do-it-yourself type of seminar. So let's get started. We have our agenda. We'll be going over what is cellular target engagement and going over some key aspects of the assays needed. Then we'll dive right into the specific tools, and now these are in-house tools you can purchase. They're vector-based tools to create your own target engagement assays. In this section, I'll introduce you to the technology behind this, the InCELL Pulse technology. Then we'll dive deep into the experimental designs and workflows. So this is the main point of the presentation. And then end up with a few applications and a case study focusing on kinases, methyltransferases, hydrolases, etc. And then finish up with a summary slide and Q&A. All right, so small molecule drugs in general, they work by acting on protein targets and eliciting a desirable phenotype. So this is a core concept of pharmacology, and many times it's very difficult to get at. So, wouldn't it be nice to determine if your specific compound directly engaged to your correct target in the cell? So we have a picture here showing you the basic concept of cellular target engagement. It's a very simple concept described in the cartoon or the image. It's a small molecule compound. It's not looking at biologics such as antibodies or large proteins, because the small molecule compound needs to be able to enter the cell and then bind or engage to the intracellular protein target of interest. Historically, though, in general, this has been difficult to measure correctly, without functional type of assays or phenotypic assays, and current biochemical type of assays for target engagement typically lack the ability to determine whether that compound, can penetrate through that cell membrane. So also in general, compound binding in the majority of cases causes a change or alters the thermodynamic stability of that protein. So binding will actually change the observed steady state levels. And what I mean by steady state levels, it's the change between the degradation and the synthesis of your specific target you're looking at. So in most cases, the binding will allow it to increase the levels of target protein due to this possible stabilization effect. All right, so let's bring this into context to the drug discovery world. You're looking at different approaches here in the drug discovery continuum. And where would target engagement play a role? So first of all, target-specific approach. This is the top portion of the slide. You start out with a target-specific biochemical screen, for instance. You have like 500 targets, or excuse me, chemicals that you want to look at. You identify them, and then you follow this up, show or prove that they have some kind of target-specific cellular activity. So you may use cellular assays like a functional-based ones, or maybe an indirect phenotypic assay. This is also a nice place for target engagement assays where they would be applied. And then once this is established, compounds can then be tested in a more complex cellular animal model to validate the target and the compound drug-like properties. The second approach, a little less common, but you may start out with a phenotypic screen, perhaps a cell-based assay screening against the compound library. You identify hits that give you that specific desirable phenotype. Then perform target convolution studies, which sometimes can be very difficult, but you want to answer, what is the target of my compound that is giving me this specific phenotypic or this effect? And this tends to deliver some really good hypotheses, which then obviously need to be tested and validated, and that's where target engagement could be introduced. You prove that the compound will actually engage with that specific intended target in the cellular context. And then this data will help validate the target in general. So these are two key areas where cellular target engagement assays can be applied in the drug discovery continuum. All right, so now getting back into the lab. So we are a customer and we would like to use a target engagement assay system. So what are the needs of our customers? The main need is obviously, is my compound cell penetrant? So that's the first bullet point in the blue box or the indented one. So it ensures your compound gets into the cell, and they want to show that the compound actually binds or engages with the specific target that's intended. You may also want to rank, order how potent your specific compounds are against the target. You may want to confirm target engagement for a specific target that you believe is responsible for a particular phenotype of the cells that are treated, as I mentioned on the last slide. But overall, the specific assay requirements is customers want something quantitative, easy to use, and an in-house type of solution that's very flexible, ability to do high throughput type of approaches. They definitely want it low cost and very generic. They want to use their own targets and obviously compounds without any need for custom reagents, and obviously give you accurate results. So those are the main requirements or assay needs. And DiscoverX has wanted to create a solution for all these assay needs, but it first started out with a few years ago, there was a breakthrough with the introduction of cell-based thermal stabilization assays, and these are called thermal shift assays. Their powerful concept of thermal stability changes, like induces the ligand binding to demonstrate that the ligand is actually bound to the target. So let's look at the first graph, the lower left-hand corner. This is a concept of where your cells are actually being heated at increasing high temperatures. You're looking at the black curve in this first graph, and that's showing you the target protein actually denatures or aggregates as the cells are being increased in temperature. But if a ligand is bound, for instance, this is now the red curve. So the ligand goes into the cell and it starts to be heated with the system, temperature required to denature the protein. These proteins are actually, the temperature is shifted to the right, so the proteins can withstand a higher temperature. Now this concept can be exploited to then use an isothermic type of approach as a function of the compound at different temperatures, or different concentrations and measured at inhibitor potencies. So the current methods, they may take advantage of traditional type of technologies, a Western blot, ELISA, like mass spec type of approaches, where they may require custom antibodies. Some other approaches may require chemical probes, et cetera, to evaluate. But we've introduced at DiscoverX a new technology called InCELL Pulse. And this does take the current thermal shift assay type of technology into account. And this new product uses engineered cells. The assays definitely are very simple and rapid, easy to use. They're also homogeneous, so no wash steps, unlike typical like ELISAs, for instance. They are generic, so you can use this technology to evaluate a diverse set of targets. They're high throughput and quantitative, and the readout is very simple. It's just a chemiluminescent readout, allowing you to measure this target engagement without the needs for antibodies or mass spec or chemical probes. All right. So overall, you will obtain melting temperature, shift data, isothermic experiments will be performed, you'll get EC50s, signal to background noise or ratios, etc. So let's move into the tools to help you create your own target engagement assays. So introducing you to the tools, I first wanted to show, what is the technology that is behind InCELL Pulse. And you may have heard of this. It's from DiscoverX from the early 2000s. It's called the Enzyme Fragment Complementation technology. And basically, it's using a B-Galactosidase enzyme. This enzyme has been broken up into two fragments, a very small enzyme donor fragment. This is that little red squiggly line called pro-label. So think of this as your enzyme donor. And then there's a larger fragment called the enzyme acceptor. Now that's that gray structure in the middle of the slide. Together they form an active enzyme, but when they're apart, they're inactive. And this allows you, when they're inactive, you can take your small pro-label and bind it to your target of interest. And then with the addition of the enzyme acceptor, these two now can combine and form an active enzyme, and then with the presence of a substrate, which is hydrolyzed, and then this produces a chemiluminescent signal that is read out and gives you a nice signal on a dose-response curve. So in general, the small peptide, it doesn't interfere with your protein at all as far as structurally. It's very sensitive detection for properly folded target proteins. You can think of it as like a biochemical tag that senses the folding state of the protein. So for instance, if the protein is denatured or aggregates in any way due to temperature, in our case, this causes for poor complementation. They don't bind together and results, the readout is reduced or decreased chemiluminescence. All right, so if we add one more variable into the scene, we're going to add the heat denaturation part and put it into context of the cell. First, you start out with your engineered cell line, expressing your protein target tagged with a pro-label, that little red squiggly line. And then you introduce your compound to the cell and perform heat denaturation. This is done by a PCR machine. And this allows you to customize the assay parameters. How many seconds or minutes do you heat it up and at what temperature, et cetera. We'll go over that during the design of the experiment. All right, so now if the compound that you introduce does not bind, so we're looking at the lower right-hand part of the image, there is no target engagement, and that protein denatures and aggregates, complementation is inefficient, and you get that reduction of the signal. So less of the small pro-label tag binds with the large EA or enzyme acceptor. Now if the compound does bind, you're looking at the upper right side of the image, there is target engagement, and this is due to protein is stabilized and precludes the aggregation. Thus, with the introduction of the EA and the detection substrate, complementation occurs and you get the chemiluminescent signal the readout in a dose response type of manner. All right, so what are the tools you need? As I mentioned earlier, it is a vector-based product that you can use to make your assays in-house, be creative as you'd like. The assay to develop this takes less than 2 weeks, but this is of course after the cloning step that is required. You have complete control over the cell line that you want to use. Use your favorite one, U2OS, CHO-K1, HEC293, etc. Then of course you choose the protein of interest that you'd like and the compounds of interest. And then you design it. Transient transfection works well. No need to build stable cell lines at the beginning before you're developing your assay, because all the testing is done just prior to that. What comes with the product? This is called a starter pack or a starter kit. Includes the vectors. So those are the key components. You have 2 different pro-label vectors, so you can tag your protein at the N or C terminus with this pro-label tag. You have a positive control vector. This is called MTH1, and this is just a hydrolase. And we'll go over how this is used in the next couple of slides. You should definitely run this in parallel with your experiments to make sure everything is working properly. Then you have the detection reagents. These are the EFC reagents, including the substrate, enough to allow about 400 data points. And you have the plates. We provide those that can work both in the PCR machine for the denaturation step as well as the luminometer. And then control reagents are the ones to help you ensure your assay is optimized. All right, so let's now move into the experimental design. We're looking at a flow chart on the right of how to do the assay development steps. This is just a high-level overview. First, starting with the cloning and transfection steps, so that's sections 1 and 2, and then moving into the main assay development sections. So there's 3 different phases. I'll go over those in the next couple of slides, and then finally ending up with the creation of your stable cell line. All right, so let's get going. We're going to talk about the experimental design and workflow and go into these steps in a little more detail. All right, so the first phase, let's see, it's cloning and transfection. So the goal of this step is to add the EPL or the pro-label tag to the C or N terminus of your target protein and expression, construct, and transfect. So we definitely provide the vectors for this. You could take your target, it could be full length, or it could be just a specific domain. For instance, the catalytic domain binds, the ATP domain binding site of a kinase, for instance. You want to do the cloning step and then the transient transfection, which takes place over about 3 days. Plating, transfection, and harvesting. And here you're looking at an example of one of the vector maps. The cloning will obviously be done at the MCS site or multiple cloning site, and you see there is a EPL tag there with a small or a short linker, TEV, between them, and that allows you to do the transfection. All right. The next phase gets into the main assay development phases. So there's 3 of them. This is the first of the 3 that you perform after the transfection. And the goal of this is to basically determine the variables that will be used for your EFC-based detection part of the assay. So basically identify the conditions that will be used to ensure you get a very high signal to background ratio. And the protocol kind of shows that flows as followed on the slide. You want to obviously design the plate layout, do the preparation and harvesting of your cells, and assemble the assays, multiple EFC detection solution conditions set up basically to test multiple conditions at this point. So that's why it's in blue. It's the most important part of this, this step. Then you want to perform the InCELL Pulse detection reagent or reaction. Read it, analyze the data, and then based on this data, you will select the specific EFC detection solution conditions, and these variables will be held constant going forward. We have some guidelines also in the user manual that is available online if you would like to read it at any time. I'll have a link for you at the end. All right, so let's move into the second phase of the assay development process. The goal of this is to identify thermal denaturation temperatures. Okay, so you want to show that your ligand is bound to your protein and it makes a shift to the right, indicating that it is bound. So the first step is to perform, the melting temperature curves for the protein target generated in the presence or the absence of your inhibitor. And so you can kind of look at this figure on the right. It is the same one that I showed earlier. It was much smaller then. So this is much easier to read. You see now that you heat your cells at increasing temperatures, for instance from 37 to 60 degrees Celsius, without the ligand, that is the black curve. The amount of EFC signal that we get decreases pretty steeply as a function of the temperature due to the denaturation or the aggregation of the target protein. And therefore EFC is becoming less and less efficient. Okay, then next you add your ligand, and this binds to your target. You're looking at the red curve now, and you see a TM shift occurs. You see that horizontal blue line in the middle of the graph. And then you see that the target is it does denature, but at higher temperatures this time. So at any given temperature, for instance around 50 degrees Celsius, there is an assay window. And this is your signal to background ratio. So, if you titrate your compound in, looking at different concentrations, you can start with as much signal and as a function of the compound concentration to get to this specific highest signal. So again, the goal is to use a known potent inhibitor at this step. So I suggest use an inhibitor that you identified by a biochemical screen, for example, and that will allow you to get this TM shift. And there's also some control experiments that you will perform to evaluate the compound's effects, and the details again are in the user manual for that. All right, next step. Oh, this is just the same phase 2, but it goes over the protocol steps in a little bit more detail. It's very similar to the last phase 1, except for preparing the positive control. You want to stick with a single concentration at this point. And then during the setup, you want to treat your cells, with the compound as well as, a DMSO type of control. Don't forget that. Do a range of temperatures. For the pulse denaturation step, the thermal gradient that's highlighted in blue in this slide, you want to use obviously a thermocycler or a PCR machine. Set up your plate, one plate added to one PCR machine for a single run. Your cells are in suspension with the compound at this time. You perform the detection reaction and then analyze the data and choose the temperatures for the next step. So that's the main goal. So in this graph, you see that we looked at the control vector that was created. So MTH is bound to pro-label, this hydrolase. We ran the experiment, we want to run it in parallel with the one that our target of interest. The black curve is the DMSO negative control, showing the target is denatured, has occurred, and then the red one is using a known potent positive control inhibitor. And this is the TH588, and the TM shift definitely is to the right, and it shows very good separation between, the signal to background. All right, so the goal again is to choose the temperatures for the next step. And just some guidance here, you'd want to choose the lowest temperature and then a couple of flanking temperatures around this, and that was acceptable. And sometimes if you choose the high temperature, this affects the apparent affinity of the compound. All right, so now here's phase 3, and the goal of this phase, is the final step of the main assay development process, is to measure the isothermal inhibitor dose response curves. So now we're looking at just 1 to 3 different temperatures as determined by that phase 2 step on the last slides. So for instance, you're going to be looking at maybe 47 degrees to 57 degrees, a very small window. Design your plate, prepare the positive controls, and now this time instead of a single concentration, you're going to do serial dilutions. And you dispense your compounds to the plate, harvest and transfect your cells, set up the assay, and incubate with the compound. And then the main step is performing the isothermal denaturation type of experiments using those different temperatures identified prior. You're going to look at these dose response curves as a function of temperature to determine the final conditions. So this would be, okay, let me look at the IC50s at this point, as well as the good assay windows. Do your detection reaction, your readout, and analyze the data and choose your final conditions. So after you're done with this, you're finally happy, the main experiment went well, you can go ahead and generate your stable cell lines. Or if you want, you can definitely continue to do transient transfections to test other conditions and targets, etc. All right, so here's just some characteristics just to summarize from the last slide as far as assay windows. You really want them to be above 2 no matter what, preferably above 3 for potent, positive control inhibitors. And you want to confirm that your rank order potencies, the EC50s or IC50s for your known inhibitors are correct. So again, here's looking at the control, the MTH1 example. And you see that the EC50s are nice in a good order that corresponds to the results seen in the literature for these specific compounds tested, and the assay windows are all above 4, so it's very nice, obviously, except the control inhibitor, which is a lipid kinase. The post-denaturation steps for this specific target was about 2.5 minutes long at 48 degrees. So that was the condition used for this control. All right, now I'm going to move into the last section of the talk. This is the application and case study section. The applications were, kind of mentioned at the beginning of the talk, and they're kind of summarized here. So obviously you want to establish cellular permeability, something that is not done through biochemical type of screens. And you want to obtain, target engagement of your protein, of your compound of interest. You want to verify cellular activity from hits from, your biochemical screen. Rank order the compound cellular potencies, help. This is during your lead optimization type of steps. Analyze the targets where they may be lacking a simple or direct type of functional assay that obviously may not be available. They're kind of hard to come by for more unique type of targets. You want to establish the mechanism of action for your compound. That was maybe a hit from a phenotypical screen. For instance, a hypothesis of compound X binding kinase Y and prove that you get cellular target engagement. Maybe you want to de-risk off-targets activities. You may have seen, your compound from a broad biochemical type of screen. And then all these can be applicable for many different targets. We've tried hydrolases, methyltransferases, and many different kinases. And our customers are trying a lot more, and we'll keep adding this to our collection to help you with your project. All right, so our first example is looking at ABL1. Now this is a tyrosine kinase, looking, maybe involved in leukemia. And what's really nice is you can characterize different types of inhibitor classes. And what I mean by that is, for instance, type 1 type of inhibitors, they bind to the active conformation of this specific target, ABL1. Type 2 type of inhibitors bind to inactive conformations. Then maybe you're looking at non-ATP competitive allosteric type of inhibitors. So allosteric inhibitor means it binds to a non-active site location but still affects the binding. Let's see. So here we have dasatinib, which is a highly potent type 1 type of inhibitor, known with cellular activity within the single-digit nanomolar. So we recapitulated this from the literature to show that it worked. Imatinib was another. It's a first drug approved for this specific target, and this is about in 100 nanomolar activity range. So we showed this. Then we looked at GNF2 compound. This compound binds outside of the active site, so it's an allosteric, non-ATP competitive effects going on. It actually binds to the myristate type of binding site, so the non-ATP binding site of this specific kinase. And then we have our control inhibitor, the lipid kinase that was used in the earlier study. Now ABL1 as well as that MTH1 hydrolase We already have those available as kits ready to use. We already did all the cloning, transfection, et cetera, stable cell line generation for you. If you'd like those as a test case, we do have those available. All right, so now we're going to look at analyzing challenging targets, and that MTH1 hydrolase is definitely a challenging target. Its interest is also in cancer. Inhibitors basically affect the cell proliferation, and there's a lot of difficulties building functional assays for this one. And earlier thermal shift assays were used to prove that the inhibitors of this enzyme were actually cell penetrant and engaging. So we wanted to make sure we recapitulated this type of study. And then we have our G9A. This is our first methyltransferase we're looking at. It's an epigenetic target. Definitely difficult to create functional assays for these. And the readout actually could be very complex and sometimes requiring mass spec. So it's nice, to have a nice, convenient, simple cell-based target engagement assay that you can use to answer these questions. All right, so those are difficult, challenging projects. Now we have a case study. So DiscoverX is working with the Structural Genomics Consortium. This is based out of North Carolina's site. So the SGC is focused on working on developing potent and selective chemical probes or inhibitors for every kinase in the kinome. So, and they do realize there's about 500 plus, kinases, and only about 15% of those have dedicated, well understood inhibitors. So overall, this is a potential gold mine, of remaining 85% of the relevant targets for diverse disease indications. So by determining these chemical probes or inhibitors will definitely pave the way for this specific initiative. And proof of the target engagement is a key specific step in their strategy, as shown in the flow chart. So what they do is they start out, working with pharma collaborators, get their hands on as many diverse inhibitors as they can, assemble the set and maybe screen it over using a biochemical type of a screening approach. They worked with us using our Kinome Scan biochemical service here at DiscoverX. They identified compounds of a 500 or 600 molecule library for which kinase targets do I have that are potent and selective inhibitors. So they really did focus on understudied kinases. They wanted to, ultimately ID them and prove the molecules and then test them ultimately in a phenotypic type of screen. When kinase targets are good for specific diverse disease indications. So in general, though, the phenotypic screenings can be very expensive and lengthy. So before they start this step, they wanted to find, does my compound get into the cell and engage to my target? So this was a key step in helping them reduce the number of molecules they moved into the next phenotypic screening step. So let me just show you two examples, GAC and HASPIN kinases, and look at their potency rank orders that they established. And this is shown in this slide. The GAC kinase at the top, HASPIN at the bottom. They successfully ran the assays, generated the rank order potencies, obtained sub 100 nanomolar type of levels of concentrations, or EC50s, excuse me. Left to right, it gets less and less potent. So hopefully this shows you the importance of doing cellular target engagement, especially with kinases, not only determine if the compounds can penetrate through the cell, but also looking at it as far as a competition with the cofactors for kinases specifically, because there's ATP. So kinases all bind ATP, generally competitive with respect to that inhibitor in most cases, and kinases have different affinities for ATP, so keep that in mind. So when you're comparing your data, your biochemical data with your new cellular data, there could be a 10 to 100-fold offset difference between these type of data, so just keep that in mind. All right, so that was our collaboration with SGC. Now we have a GSK poster. These two posters shown here, though, in general are from the SLAS conference. The GSK poster focused on using cellular thermal shift type of assay. It's from called CETSA, and they introduced the InCELL Pulse technology within their experiments, and they made it like a high throughput 384 well type of format and looked at many different diverse targets. So I highly recommend to look at that. The link is at the bottom, which I can definitely share with you later. And then we have our poster that we've done at the SLAS conference as well. Most of the data, if not all, has already been described in this talk or available on our website. But I wanted to focus on a small portion of the right-hand poster where it has a small circle on it. And I'll blow that up, and it's shown here. So what we did was we took the kinome and we wanted to spread it out and create, look at all the targets throughout the human kinome that kind of balanced it out, this specifically the human kinome. And we created about 25 different, we looked at 25 different kinases as shown here in the table. And then we put together this as a package. So if you need any of the EC50 data, SB, looking at what compounds did we use, we have this data for you in a reference set. The kinases obviously include understudied kinases as well-studied ones. And common off-target kinases definitely were studied, and these were identified by biochemical inhibitor selective screens. So we have this data for you, and if you need the expression vectors for any of these specific targets, we can make that available to you as well. All right, I'm going to end the talk with a quick summary and then move right into the Q&A session. So hopefully I was convinced you, the role of cellular target engagement, and obviously in the drug discovery process is ideally very important for establishing, whether your compound can penetrate through the cell membrane and engage with your specific target in the cellular context. We have a solution for you. It's easy to measure, single or simple readout, binding with a chemiluminescent type of readout as well. No custom chemical tracers or antibodies is needed. This provides a convenient solution for you. It's definitely a more robust than many of the solutions out there, and it is in a product form that's very generic for a diverse set of targets you're interested in. This is especially important if there's no functional assays that are available. And it definitely complements biochemical assays, if you want to de-risk off targets identified in these screens, for instance. And then obviously you can use it for high throughput screening as well. All right, so thank you very much, and I will begin to take any questions that you may have. Thank you.
Thank you, Dr. Haley Vicente, for that informative presentation. It's time for Q&A. If you have a question you'd like to ask, please do so now. Just click in the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the Send button. We'll answer as many questions as we have time for. If we are unable to get to your question, you will receive a follow-up email after the presentation. So let's get started. Our first question is, what is the most important step during the InCELL Pulse assay development?
Oh, thank you. So the most important step is probably looking at all the different variables. You want to focus on the thermal denaturation step as being highly important, obviously. But I would say the key step is the cloning. You definitely want to try multiple constructs for the highest success rate. You want to carefully, obviously, choose your reference inhibitors. And you don't want to skip the phase 1 assay development part, specifically running the positive control. So again, designing your constructs carefully. Make sure you put that PL tag on both, the N terminus and then try the C terminus. Don't skip optimizations for readout conditions. So you get sensitive and you want to get a true readout. Next question.
Thank you. So the next question is, how well does your system work on full-length versus domains? Any examples of full-length proteins?
Oh, yeah. So I mentioned earlier in a slide that they definitely work for both full-length and domains. We've tried both for a lot of the kinases specifically. So again, you should test both. Make your constructs using the full length or the domains. This is definitely that critical step. Maybe focus on a catalytic domain, obviously when it is a full length protein. For multiple domain proteins, these are these can be difficult. So definitely try a domain. This is a good example of this. Is that ABL1 kinase. I hope that answers your questions. Okay, next one.
Thank you. Can InCELL Pulse be used in higher throughput formats like 384-well?
Yes. So that GSK poster was a nice example where they took the Pulse technology and they were able to adapt our 96-well protocol to the 384-well format. So yeah, this is definitely applicable, and it was like 3 slides back. Okay.
Okay, great. Okay, and we'll wrap up with one more question, and that question is, have you tried to look at membrane protein targets?
So that's a good question. So we have several customers who are looking at membrane proteins, and we do have this under development in-house as well. It is such a new technology. But we assume that this will probably work because there are examples of thermal shift assays that actually, look at integral proteins. For instance, the sodium-potassium ATPase pump was done. The drug transporter, that MDR1, was done. We also assume that some NHRs would probably work really well with this assay as well. So definitely, try it, and obviously our tech support team can help you get through some troubleshooting spots if you need to. Okay.
Great. I would like to once again thank Dr. Haley Vicente for her presentation. Do you have any final comments?
Yeah, just if you have my email here, feel free to email me additional questions that I can answer directly to you, or contact our technical support. And again, we have a web page also indicated at the top of the screen for InCELL if you want to learn more details. And thank you very much for joining the webinar today. Thank you.
Thank you once again, Dr. Haley Vicente. I would also like to thank LabRoots and DiscoverX for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through November 18th, 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.